And so what I've been working on is a very targeted approach using what's called shear stress, which is the frictional force of blood flowing through these vessels, which through my preclinical models I found is much, much higher in these vessels than anywhere else in the body. Hi, my name's Daniel Beard. I'm a senior lecturer at the University of Newcastle in New South Wales, Australia. Also a visiting scientist at the University of Oxford in the UK and the Chief Scientific Officer for the company ShearFlow. My educational career and journey started, I guess, at high school. I was always interested in biology and how the human body worked. Probably wasn't the best student, but I was fortunate to have a really inspirational and motivating biology teacher that encouraged me to pursue a career in medical research. The next, I guess, turning point was through that study of medical research. I was always fascinated with how the cardiovascular system worked, but also how the nervous system worked in neuroscience. And that, I guess, naturally led me to pursue a career in stroke research, where a blockage of the blood supply, the cardiovascular system, leads to neurological dysfunction. And that led me to pursue a PhD in Human Physiology, which I graduated with in 2015, which led to, I guess, my next turning point. I was really fortunate to secure a postdoctoral fellowship at the University of Oxford to work in one of the leading sort of neurovascular research labs of Professor Alistair Buchan, and pursue a really exciting time of my life, living 3.5 years in Oxford, which at that time I was fortunate to set up a collaboration with Professor Donald Ingber at Harvard University, where we set up a collaboration to look at a nanoparticle therapy that can boost blood flow to the brain during stroke, which just before 2020, when the world went a little bit crazy, my wife and I moved back to Australia and was fortunate to secure an NHMRC Ideas Grant to establish my lab back here at the University of Newcastle, where we've been continuing to develop this novel therapy for stroke. My main research focus is in the preclinical stroke space, so using preclinical models to study the pathophysiology of stroke and using that information to understand and design therapies to improve stroke outcome. And as I mentioned, my research interest is mainly in the cardiovascular system, so I'm a bit more of a pseudo-neuroscientist. I'm more interested in the pipes than the wires, and this has led to some of the work that I've been doing looking at these really interesting blood vessels in the brain. So when you have a blockage of blood supply to the brain, fortunately our brain has this built-in network of collateral vessels or bypass vessels that can redirect blood flow around the clot. And that gives rise to an area of brain tissue that's dying but not yet dead, and this is known as the ischemic penumbra. And there's been, over the last 10 to 15 years, really strong imaging studies, so CT angiogram, CT perfusion studies, a lot of the groundbreaking studies that have been done here at the University of Newcastle in Australia that have shown that the better the blood flow through these vessels, the better the longer this tissue, this salvageable tissue can survive, and the better the patients do. But one of the real challenges is opening up these vessels or making more flow through them is quite challenging. We have lots of drugs that are available that can open up vessels, vasodilate them, but unfortunately, if they're non-targeted, they'll go everywhere in the body, and that can cause systemic side effects and could potentially make stroke patients worse because you drop blood pressure or redirect blood flow where it shouldn't go. And so what I've been working on is a very targeted approach using what's called shear stress, which is the frictional force of blood flowing through these vessels, which through my preclinical models I found is much, much higher in these vessels than anywhere else in the body. And in collaboration with Professor Ingber at Harvard University, who had been producing shear stress sensitive nanoparticle aggregates, we could package vasodilating drugs into these nanoparticles and selectively deliver them to these collateral vessels to sustain the penumbra for longer and hopefully improve stroke outcomes. And we've got some really exciting preclinical evidence that we've just published in Advanced Science showing that that is the case. My biggest collaboration has to be with Harvard University. I've been working with them over the last 4 or 5 years to develop this nanoparticle technology to improve brain blood flow. We're continuing this academic collaboration into a more commercial collaboration with a spin-out called ShearFlow, where we're now commercialising this technology for stroke treatments, and it's a joint venture between the University of Newcastle and Harvard University. So that's really exciting. That has been funded by, this work has been funded by the National Health and Medical Research Council of Australia through an Ideas Grant. I was really fortunate to secure that when I moved back to Australia, which allowed me to establish my lab and move forward. I've also been really fortunate to secure funding from the Brain Foundation, the New South Wales branch, to do further safety evaluations on our technology, and that's gone really well, and we're really excited to share those results soon. And also really happy to be supported internally by the Hunter Medical Research Institute Heart and Stroke program, as well as the University of Newcastle School of Biomedical Science and Pharmacy internal grants and equipment schemes that have allowed us to acquire some really cool blood flow imaging equipment that's allowed us to assess blood flow in our stroke models. Curiosity and a need to make a difference, I think, is probably the best way to summarise it. The curiosity, I think, came from my uni days studying from textbooks, and I just loved, just absorbing as much knowledge as I could, understanding how the body works. But then it was a professor of mine that said, well, textbooks are great, but research is where it's at, because that's where you're going to be creating new knowledge. And that really struck a chord with me and got me going down the research path, and always sort of chasing that really awesome feeling you get when you discover something unusual or really cool, or something that could make a real big difference. And the need to make a difference, I think, although my academic interests led me to stroke, it is a huge unmet need. 1 in 4 people alive today are set to have a stroke. Only 15% of patients currently receive therapy, and only 50% of those achieve a favourable outcome or benefit from those therapies. So the therapies that I'm working on will hopefully improve those numbers, save brain and reduce disability for stroke patients in the future. So that is my sort of massive transformational purpose that I really want to keep pursuing. Spending time with my family is probably the thing that I enjoy the most. I've got 2 young twin boys, 3 years old, so they keep my wife and I very busy. So outside of work, I try to spend as much time with them as well. I'm very proud dad. But between work and family, it can get a bit hectic, so I do try and eke out time for running. I've just completed my first marathon, the Gold Coast Marathon, in July. It was an amazing experience, although I had to get up at stupid o'clock to make it work. I really enjoy running. It helps me sort of start the day right, get my head straight, de-stress, as well as all the other cardiovascular benefits that comes with it. So yeah, really enjoy doing that as much as I can.